Hello and welcome back to the Computational Genomics Lab. Today we are going to start to explore our results. We've collected them all into one data frame, and now we're going to do some real genomic analyses and say which genes are significant, which are changing across time, what does base mean, and these kind of things to learn some of the fundamentals of RNAseq, some basics of R, all while analyzing our data. Okay, so we're going to just go here on the GitHub. I'm probably going to start to forget to do this because I think by now you probably already found this. You go to Masterclass, Lessons. We are on 06, Differential Expression, and today we are going to do 04, Exploring Our Results. And so you can have your own local setup on your RStudio on your local computer. You could be on a supercomputer, but really one thing you can do is just hit this raw button, copy and paste this into RStudio by making a new R markdown file, and you'll have the exact same thing we're doing here. Okay, so I've loaded my libraries here, and now we are going to load our time point ResDF. That's what we made last class. That's had the for loop that went through and grabbed all of our results. So it's in one data frame. There it is. Okay, so now one of the first things you want to do is say is which of my genes are significant. And remember, this data table with 221,000 rows has all the genes in all the time points. And what we're going to do now is start to say, well, tell me if in the p-adjusted column, which ones had a p less than 0.05, the kind of standard lowest statistical thing you want to do. Now, granted, that is subjective, and you can make that 0.1 if you were interested in it. But for now, we'll just go with the standard. And so we're going to make a filtered ResDF, which this now should shrink quite a bit. And we're going to pipe in ResDF into tidyverse pipe here, and then just run the filter function of p adjusted less than 0.05. Okay, let's see what happens. Voila, we can see we're down to 14,000 observations from 221,000. So a lot of genes, again, aren't even expressed or are going to be significant. And then the next thing we want to consider is not only if it's significant with the p adjusted, but also how much it changed. And so in log-fold 2 space is really nice because log 2-fold of 1 is 2-fold, and log 2-fold of -1 is 0.5 or half down. So it's very symmetric, and it's what we're going to use. And so we're going to say the absolute value. We're going to do the same thing. We're going to take our filtered ResDF now because we've already done the 0.05, and we're going to do filtered p-adjusted 0.05. Nothing's going to change there because we already did that. It's more just to show you in the filter function in R, you can do more than one filtering at the same time. And so we're going to say ABS for absolute value of log-fold change greater than 1. So that means it either went 2-fold up or 2-fold down. 1 and -1 will both pass this. Okay. Let's try and see what happens here. So we're currently at 14,000 observations. Let's see how much this shrinks it. Wow, 918. Okay, so now we're talking real gene expression differences. These are genes that had changed in at least one time point. We don't know which ones yet because that data frame had all of our analyses. And we know that they changed at least 2-fold in one of the time points and were significant by Deseq to 0.05. Great, you could start to have a lab meeting now and say, I found 918 genes that change at least one time point upon exposure to docs in mouse stem cells. Okay, so now the problem is the same gene could be counted at least 4 times in each of the comparisons. So let's see how many unique genes there are. So that'll tell us if lots of the genes were changing across all the time points. It's probably unlikely, but it's just important to remember that each gene has 4 entries, or could have up to 4 entries, 0, 12, 24, 48, and 96. Okay. So we're going to use the unique function in R, and we're going to call this object siggenes. And we're just going to take our filtered ResDF gene ID column and say, tell me how many of those are unique. So we have currently 918, and let's see, and we get down to 669. So that means a few genes are showing up more than once. And we'll get into code that could also find those. But first I want to focus on the things you would do first and have a good feel for those genes. Okay, so let's find out what our lowest p-value is. Again, we could do this simply by looking in here and say, okay, p-value, sort on that, p-adjusted. Okay, wow, negative 135. So that's super significant. This guy's interesting. H19 is a long non-coding RNA, and we'll get into that one later, but it's highly significant, and we can look over to its log fold change. It's 4-fold, so 2 to the 2 is about 4. Air is changing. PEG is a paternal expressed gene. So lots of cool things in here, and these are all significant. And everything in this list here is changing due to docs. Okay, and we can do code that uses min so we go into the filtered resdf, cache sign into the p-adjusted column, and we'll call this the lowest p-value. You could also do this with tidyverse by simply saying take this column and find the minimum. Let's do that. Okay. So we just now made lowest p-values 1.2 by negative 135, which we just saw in the table. Okay, so let's say though that's just one value. Okay, so that doesn't really tell us much. And when we looked in the table, you can do it there too. You can just look through here. But if we want the whole row, we have to give it some more code. So we're going to go call it lowest p-value, but we're going to get the whole row, so we'll have base mean and all the other stuff with it. And we're going to take filtered resdf2, which is what we made when we filtered by 0.05 and log fold 2, not the unique thing, just the double filtering. And we're going to say which.min is a function to say which is the minimum. In filtered res df2 column p adjusted, give me all those rows, give me only the rows that pass this, and then all the columns. So we're going to retain all the data. So now we'll call this lowest p-value, and now it's an object in our environment. It's got one row because we did which.min, not a filter, and it's this gene we saw before. And it's high. Oh, this is cool. So like we now know this gene changed the most at 48 hours. And we're going to start to learn more and more about these genes and when they're changing. Very exciting. Okay, so now let's find the max fold change using code. Okay, if we do it this way, we take our resdf2 that's been filtered and we look for the log fold 2 column with the cache sign index and find the max. Okay, so we get a max value of 2 to the 20th. We saw that in that figure before. That's a massive change. But if we wanted the whole row, we can say, take this data frame, index with the square brackets, which.max in this column, give me those rows, and then all the columns there. And so the gene with the biggest fold change is GM9045. So you can Google what is that gene. This is where it becomes really fun. It's probably a long non-coding RNA with that name. So there's protein coding genes in here and non-coding RNAs, and you can sort and find out. And we also see that this one's got its biggest change at 12 hours over here. So there's a lot of handy information in this data frame. And now let's see all the genes that we're going to consider significant by 0.05 p-value, p-adjusted, and log fold change of 2. We can paste this out into our console down here using the cat function, which we did in Bash as well. It just prints out what's inside here. And we're going to paste out the gene names that are in our filtered df2 by 0.05 fold change of 1, absolute fold change of 1, and then collapse it with a new line with this comma separating that part of the a different parameter in the function paste. Okay, so we can put that and then we'll call it an object docs gene. So if we just run this here, it's going to print them out in our console, and you could copy and paste them from there. But now we're going to make an object called docs genes, and we can see, yikes, I don't know why that is empty. It must be in here. It says docs genes null. Okay. Ignore that. I think this was more for printing it out onto your console with this part of the function and not storing it as an object docs genes. Doesn't seem to work. But we can do as.dataframe. Maybe that was the reason I had it in here, to show the difference. Don't know. But as.dataframe will certainly work, because we're going to make a generic Excel file and we're going to say make it just this column, the gene name column in our filter database, and collapse with a new line. You could make it a comma as you wish, and you could do question mark as.dataframe to find out what other ways of collapsing there are. Okay, so now let's do that. Okay, we have a docs genes here, our 918 genes, and we only have their name. So this is like if you wanted to do Go analysis, you could paste it into enrich_R, which we have the website in a previous class is the one I like to use, and see what common pathways there are. But now we have it in a handy sort of data frame format, and then we could unique it again, and this should be that 600-ish number. Okay, yep, we rewrote over it. Notice that, how dangerous that can be. Know what you did to get to there. Okay, so I think that's really all we need, and I think docs genes here has got this funky column name because that's how we made the data frame, by indexing there. So it was like, hey, I'm just going to call it that. But you could rename it. Here is just some code to do that using the rename function. So we're going to say gene_name equals that weird name there. So it's kind of odd. You're saying put this name for that thing. And do that, and now if we look at our docs genes, we have a nice handy gene name column. That will come in handy quite often. It's pretty easy. You could also use mutate, which we did in the previous class, but let's save it, because we just did a lot of work to get to that. So that's our significant gene list. Moving forward, that's what we're going to work with. And so we can write this out in a different way with write.table. You can pick which one you like to use. There's many ways to do this. Write.csv, right? So write.csv2, you can Google around, but write.table is pretty base R and a safe way to do it. And so this is kind of cool, I think. So we're going to say, take this object, docs genes. It only has one column, so this is not super special in this instance, but only take out the gene name column and say row names are false, call names are false, and print this as a gene_name.csv. So let's make sure we're in the right directory. So here I am. We're going to go to lessons 06. Always check before you save, because it'll save wherever you were last. 06 differential expression. Cool, this is where I want to be. I have a results folder already made. I'm going to set this as my working directory, and if you didn't have a results folder, you could just make one right here. Okay, so let's do that. Voila, and if we look in our results, we have a gene name CSV that was just made today, and we can view it, and there we go. So now we can drag this using our file transfer program, copy and paste it into a web browser, and start. Oh, I even have the website here. So I take this, go to this site, and try taking that with a file transfer program, bring it to your desktop, opening it up, copying and pasting it, or copy and paste from your console if you'd like down here, and see what pathways are enriched. Okay, so there's some other things we want to consider in RNAseq, and one of them is base mean. That's telling us about how much the gene was expressed, and you might want to filter on this level. We're going to get to that later with the TPMs. Will be something we'll also talk about, which is transcripts per million. There's lots of ways to get a value of how much a gene is, but there's also some caveats. And so I think you always want to look at distributions. So that's this hist function here. Again, level 7 is all about plotting. We're going to make much prettier plots, but hist is a base R way of saying, show me what my data is sort of looking like. And so we're going to go into our res df2, which is our filtered ResDF. Why don't I see it here? Oh, it's in here. Okay, yeah, filtered ResDF is this 918, and we're going to go into let's see, filtered ResDF2, and we're going to go look at the base mean and see how much that varies across our data. Okay, let's do that. We're going to do a little cleaning up here so we can get back to our markdown file. So we do that. Cool. We get a figure. Zoom in. Wow. Okay, so this looks like most of our data is here. That 800 plus of our genes are almost all less than 500, and we can zoom in on that. We don't have, so it looks like one of them is 50,000. And this is, I think, one of the caveats to base mean and TPM is that it's either like you're this or you're something else, but there's very few of those other things. So almost all of our data is in here. So let's take a closer look at that. So we can just do the same hist function on this base mean column, indexing with the cache sign, and say the X limit is, C is going to say paste both of these together. You need to have this. So the xlim is a parameter in hist. You can do question mark hist and look at those. And we're going to say make it from 0 to 1,000 because it looks like most of our data is before there. We can also tell it how fine to make the bars. So let's say we want 1,000 bars in our plot. Okay, let's take a look at what that did. We're going to zoom in here. Okay, cool. That looks much nicer. So now we can see by cutting off at 1,000. Now remember, there's one way out there that our data has a little more structure to it. We can see here, and most of our values are less than 200. Okay. So let's do the same thing for p-value. We're just going to go hist filtered into our data frame into the p-adjusted column. And if we zoom in there, oh, why did that get so small? Okay, cool. Well, we see everything is less than 0.05. That's good. And we see a lot of our data is like super significant over here. Cool. So I think it's just important to always take a look at your distributions to say like, yeah, what is normal? What is far out there? So when you go and do your analyses, you can always double check how common those properties are. And then let's do log fold change and see how much our genes are changing. Zoom in on that. Okay, cool. So we did the filter of a log fold change of 1 or -1, and we can see that our data doesn't start till 1 or -1. So that's great. It's filtered the way we thought it was, and that's why this is blank here. And we can see it's pretty symmetric. And the same amount of genes seem to be going down as going up. There might be a little more going up than going down, but these are all plots you could start to bring to lab meeting or to your colleague and say, hey, look, I analyzed your RNA-seq data. Okay, there we also did, I changed the xlims. You can go back through this and play around with it, and again, 1,000 breaks just to have a little more granularity with our data. So let's see, let's do a statistical analysis. We'll get into much more interesting ones. We're still kind of learning our base R functions here, but let's say, what is the median base mean for all the genes? So here we're going to go into ResDF, which has all of our genes, and the base mean is going to be the same across the time points. So it's going to say, what is the mean base mean across all genes? And then we'll ask, are the genes that are changing have a higher base mean than all the genes? This is like your first real statistical analysis outside of Deseq2, where you're saying, in my data, are these genes that are responding to dox and stem cells have a higher level of expression than the regular gene that doesn't change? Okay, so let's do that. Base mean of all genes is 0.2, so I think we're going to be higher than that from what we saw before. And let's do the same thing here. We'll take the median of our ResDF that's been filtered and ask what that value is. Okay, base mean sig genes 33. Okay, that's definitely going to be different. So that means the genes that changed had a base mean that was usually higher than those that didn't change. Okay, so we can do a t-test on that. R is great for statistics, and you can look more into t-test here, but I have the basics set up where we need to say what are we going to compare. So we're going to compare the base mean in all the genes in the ResDF compared to our filtered ResDF here. Okay, so let's do a t-test. Voila! So we got a bunch of information here, and we can see we did a 2-sample t-test. The p-value is 0.04, so that's great. That means it is significant. And we now know that our thought that if a gene changes, it'll have a higher base mean is true for this data set. Cool. So now let's look at counts. So we looked at base mean as one way to measure how much of a gene there is, but counts is my favorite way to measure a gene because it's all the reads across the gene. So there's a lot more values there, whereas base mean is saying here's one value to represent all those reads across the gene. That salmon aligned for us. So let's load our counts matrix as we always do. It's going to have that gene column in there. It's fine for now because we're going to start using indexing, so we don't care if one of them is in character. Okay, so let's take our counts matrix and just grab out the genes that are significant. That's really handy, right? So we can go into our counts table, it has all the genes, and say just grab me all the columns for all the genes, all the rows that match my significant genes. Trust me, you'll want to do this a lot. So this is worth going back over a few times, but it's super easy. So we just say, take our counts matrix, which has all the genes, and then bracket into there and say, only give me the rows that are in my filtered ResDF gene ID, because we know the counts matrix has a gene ID column. Okay, and then keep all the columns in the counts. So we'll have the counts across all the samples. Cool. So now if we look at our counts matrix here, it's going to have, oh, this is the full one. Here's our counts docs. Okay, so these are now only the genes that were significant, and we have their count values here. They're not rounded. We did that before Deseq, and we have their counts across all the time points. Super handy to say, hey, I found a set of genes I'm interested in, grab them out of another table, and you can do that just by indexing with the square brackets and saying, give me these rows and keep all the columns. Okay. And we can unique this again, and we're going to see that there's 600, and this right now it's 918. We do this, and it's going to go to 669 as we did before. Okay, so that's just double checking that everything's looking the same. And so now what we could do is that same test. We could say, what is the average counts in a given time column, and is it significantly different? But how you measure a gene is really important. So we've looked at base mean today and counts. I prefer counts, and we're also going to use TPM, transcripts per million, in the next class. But it's important to think, what am I measuring, and what are the potential caveats? What does the distribution look like? So you could make histograms of those, and I encourage you to go play around and see what is different between your significant genes and all the rest of the genes. And there's a couple ideas here for you. And we'll see you in the next lesson, where we continue to keep exploring all of our RNA-seq results. Bye for now.